between myself and him. The purpose is to encourage you guys about stem cells, to show you how he started his journey in dentistry and moved to a stem cell as an expert in this field. So there's kind of optimism for you guys. And then inshallah he will start his presentation, and then we'll give you Q&A questions, inshallah. Okay? There's small snacks available at the back. Take your time. Qahwa, some snacks and water, please. Thank you so much. Please welcome Prof. Kamal. Bismillah ar-Rahman ar-Rahim. Thank you so much, Prof. Kamal. I would like to ask Prof. Kamal, could you please share a bit about your journey in the field of stem cell and what motivated you to focus on this? In addition, ناس تقول عنك, people are saying you are Omani. So please start your journey. Talk.
Assalamu alaikum. I can say I'm very proud that when you say that you are Omani, which is true. I said to the listener that maybe I have seen Oman before she has been born. I'm very proud that I'm one of the very first generation of Jabir Buzeri Salawiya. And that's the Jabir Buzeri Salawiya. أول الناس الخريجين من جابر بزيد السلوية. So my journey started from there. And then of course I went to the university. I went back to Khartoum University. And then I came back actually in Oman. I had my private clinic in Al-Hamriya.
Which field? In which field?
I'm a dentist. So I had my private practice in Al-Hamriya for 5 years. Before that, I was part of the teaching staff at the University of Khartoum. And then I felt that I wouldn't be able to continue as a private clinician. That's why I applied to Sweden to do my postgraduate education, my specialty, and my PhD program. I started what we call it dental implant. Look at the response of dental implants, the biological response. So that was the beginning of my story. And then I did my degree. And I don't know if you are familiar with, if you know about dental implant. Dental implant is kind of screws installed in the bone. And then when the patient comes and asks and say, please need help, I need to have dental implant. If you don't have sufficient amount of bone, it wouldn't be possible to install dental implant. Then you have to say sorry to the patient, I cannot help you, I can provide you the denture, which is not really comfortable. And then you start to think about how could we solve, how could we help this type of patients. The only possibilities is to do bone graft. And bone graft is problematic, you know that. We don't have enough spare parts in our body to move the bone from certain area to another area. And again, I can explain it more in my lecture, but again, we saw that we need to help the patient by finding other sources or other options rather than bone graft. And then gradually I moved to it. Then we saw that, okay, stem cell can be a good alternative compared to the bone graft. Then we started from there, and that is the reason that I moved from Stockholm to University of Berlin, because it was better opportunities at the University of Berlin in Norway.
Okay, the other question, beyond your current work, what other emerging areas in stem cell research do you find exciting and promoting?
I think stem cell is exciting to work with stem cell, and it's still relatively still the new field. And I will show you later on, but it's very promising. You will see from my presentation that we are able to reconstruct the bone by using stem cell rather than using bone graft. And it's applicable not only in bone, in other type of diseases or defect in the body. So it is exciting, it still requires development, it still is open for research for the coming 10 or maybe more than 10 years.
How do you see the role of interdisciplinary collaboration in the field of engineering and bioinformatics in advancing cell technology?
Absolutely. I mean, without working in interdisciplinary team, it wouldn't be possible. Because when we talk about the stem cell, we talk about what we call tissue engineering. So we are engineering, we are reconstructing the tissues or all the organs. So definitely we would need the clinician, the biologist, the chemist, the engineer, biotechnology. So in my team, if you look at my team, I have candidates from all different disciplines. So it is multidisciplinary. It cannot be done by one person. It's not possible.
What would be the challenges from your point of view working in an interdisciplinary approach?
Challenges because sometimes we talk in different language. That's I mean, when you talk to the chemist, I don't understand anything about chemistry, but the advantage that I learned from them, and that is the advantage when you're working in multidisciplinary projects or interdisciplinary projects. That is, and that's what always I say to my candidate. Please try to always get grants, big grants from the EU, from the big funding agency, not because of the funding, not because of the money, because of the networking. Then you start to collaborate with people that from other disciplines. Then you learn from each other. And that is the beauty of this.
What the challenges you face with the stem cell research from your own perspective?
I think it's still because the evidence is still requires some evidence. The cost is still rather high, of course. I will show you later on, but how to reduce the cost, because we would like to introduce the therapy as a routine therapy in the hospitals, and the data shows that it's promising approach, but the cost is relatively extremely high. And how to reduce the cost, that's something that we really should do something about.
Are you optimistic that the cost will reduce?
Absolutely, absolutely, because so far the industry sectors are not really active into the field. Because we are in Europe always, they are waiting for us, for the universities to develop the protocol, develop the whatever. If you are talking about new drug, you need to do the research, and then when they feel that it's time to take it further, then they come. Then they are able to reduce the cost.
In terms of ethics,
Yeah.
what are ethical considerations do you think are the most important for young researchers in keeping such activity in their mind?
I mean, ethical consideration is something that's extremely important. You have to always consider. And that is challenging for my field, for the stem cell research. The regulations are not clearly defined so far. We are always struggling with the EU Commission. They are changing the regulations, the rules. I can tell you that to get approval from my second clinical trial, it took me 3 years. just to get the approval.
3 years?
Yes, and because of the ethical consultation. And we don't complain because we believe that it's important. Even if it's challenging, we have to cover all the elements, but still it's important.
Is it because of your involvement with human being, or is it because of
because this is a special clinical trial, it's a multi-center clinical trial. I have to consider the regulation in each European country. It's not only Norway. So I've been dealing with France, Germany, Spain, UK, and I have to target or approach each ethical bodies, ethical authority in each country. For example, I had one experience with trust. I received very difficult questions. The French authorities, they said, Kamal, what do you know about the Nile virus? And I said, I had never heard about that. And how do you deal with this kind of group of patients? Because you will recruit patients from certain small village in France, and this village is populated by this kind of virus. So you have to consider them. And then you have to find, you have to clearly define and write how you're going to deal with them. So such kind of challenges that you have to face.
One last question, because we have the right of the position. What would be advice to give to the audience about human scientists and who want to make a difference in their
I think, research is, if research is a knowledge, you could work as a clinician. I used to be a clinician. It's completely different. It's a motivation, but it's a new knowledge. Always you gain new knowledge every day. And I would definitely encourage all the young candidates to consider the research and try to be motivated for research. So, always, even in Europe, we say that as a researcher, our income is rather less compared to the clinician. But I think myself, I enjoy it more than being as a clinician.
Thank you so much. We will allow the question later on, inshallah, from the class leaders, because we would like to start this presentation. Please appreciate him for the For food and drinks, please help yourself. It's a halal drinks, inshallah, so don't worry. inshallah, inshallah. لا بيحضروها. مين انا؟ حلو مقدمه دكتوره في حقه انا حتى احس المفروض ما انا اسوي، المفروض جينيور سينيور تسوي ان شاء الله. لا ما نزل تفضل بروف.
Okay, السلام عليكم مرة ثانية. As I mentioned, I'm delighted and honored to be with you today to share some of our experience. Take the opportunity to thank Dr. Fahad and Dr. Hisnan, the staff of the college, for giving me these opportunities. I'm always happy and positive when I come to Oman, and it's always I do like to share my experience with people from Oman. Oman is my second home country. Stem cells, today a little bit talk about stem cells. Stem cells, this is my field. I've been working with stem cells for more than 15 years now. And I think what I can say is that a promising approach when we consider regenerative medicine is the future of medicine. I can tell you, and you will see that from my presentation. My background, as I said, I'm a dentist, but I am leading what we call it Center for Transgenerational Oral Research. It's a relatively large center. It's considered as an umbrella for different research groups. And the idea is that we should provide space, collaborative space, to bring bench to chair or bed research to the reality. So we have people working with the public health, others working as doing clinical research, experimental research, biomaterial, inflammation. All of us were under one umbrella. And that is my center. We call it TOR. TOR in Norwegian is like a hammer. So just keep always pushing, the candidate to work. That's why we call it TOR. Among one of the research group is the tissue engineering group, which is my group also. And again, relatively is one of the largest research group at the university. You could see that on the left side, all these are currently registered PhD students under my supervision. Almost 11 students are doing their research now, and senior scientists, researcher, and postdoc are collaborating within the group. So we are almost like 30 scientists in the group. If you think about stem cell by itself, stem cell is an approach has introduced recently and attracted tremendous interest. We all hope that it should play a major role in treatment of incurable diseases. Just short information for those that are not familiar with stem cells. What are these stem cells? We know that we call them stem cells because they are undeveloped stems. Undeveloped cells do not look like other type of cells, and they can produce indefinite supply of themselves. So they can, from one cell, it can be divided into two identical cells. Or it can develop or generate different type of cells. And that's why they have great potential for treating different diseases. So they are undifferentiated, and they have high proliferative capacity, able to self-renewal, but also to differentiate into different directions. It can go into bone, be like bone-forming cells, or can go to the liver, make liver, or can go to the heart, cardiac cells. So they can go, that's why we call them multi-potential and multi-lineage differentiation. However, their differentiation potential is different, it varies. If you think about stem cells in the blood, their turnover is rather faster, quicker than those that in the bone. So they are different. Their fate is more or less determined by either their origin or by the environmental factors. When I say environmental factors, like for example, if you change the level of the oxygen in the body, that will change the behavior of the cells. If you apply mechanical force, you might direct the cells in different direction. So you have to be very careful when you deal with the stem cell because they are completely directed by the environmental factors. And as you could see that if they get in contact with other type of cells, again, their fate will be changed. If they get signaling molecules or signaling cell signalings or signalings from other cells, again, will change their behavior. How do they work in the body? Again, they differentiate into appropriate cell phenotype. Talking about bone, they can go towards bone-forming cells. They provide or secrete what we call the cytokines or growth factors that also would influence the endogenous cells, the cells already in the body. So if you transplant new cells, these cells can direct the other cells in the body to direct them to be differentiated in a specific type of cells. So there are different mechanisms.
when you think about the stem cells, maybe you heard about embryonic stem cell or adult stem cells. Embryonic stem cells, they have greater differentiation potential, but there are ethical considerations that not be, I mean, for many reasons that it's not easy to deal with them because they can make anything in the body. They can make a new organs with the embryonic stem cells. So, but for the adult stem cells, I mean, when you think about ethical considerations, they have more or less acceptable to work with them. When you think about adult stem cells, they are either considered as hematopoietic stem cells like blood. But here we are talking about their resources, which is the blood. And we know that patients with leukemias are more or less treated by this type of stem cells. The other type of stem cells are the non-hematopoietic stem cell, or what we call the mesenchymal stem cells, which is more or less mixed cell population containing fibroblasts, endothelial cells, fat cells.
Sorry, Prof.
Yeah, and first been described in 1980 as mesenchymal stem cells. And my focus today in my presentation today, more or less, I will more or less continue talking about mesenchymal stem cells. As I said, we consider them as multipotency. They can differentiate into different type of cells, easily to be isolated. And the beauty of these stem cells is that immune response is relatively very low because they have expressed low level of MHC and antigen and secrete immune suppressive protein. So always we are not worried when we transplant this type of cells, even from, if you consider allogenic stem cells, even if it's not from the same individual. And that is the advantage of using mesenchymal stem cells. And I'm not talking about fancy research. You could see already, if you look at the website of the clinical registry, you could see that there are already more than 1,000 clinical trials being registered. And for treating heart diseases, eye diseases, skin, joint, bone, and so it's widely being used nowadays. So more than 1,300 clinical trials already been registered when I look at it just a couple of months ago. Bone marrow stem cells are the most one being investigated. But there are some clinical trials with adipose stem cells from the umbilical cord, less clinical trial with embryonic stem cells because of the ethical consideration. Myself, my group, of course, bone is my favorite because I'm a dentist. I am leading or involved in 5 ongoing clinical trials. So I'm not talking about laboratory work, I'm talking about patient now. We already treated more than 150 patients by using mesenchymal stem cell. For jawbone, for long bone, in collaboration with the orthopedic surgeon, we have done completed clinical trial for treating multiple sclerosis with the neurologist. I'm involved working closely with the plastic surgeon to repair skin after burn, and again with the ophthalmologist that we are trying to repair the cornea after being defected. So these are already registered ongoing clinical trials. But coming back to bone, because as I said in the interview, that's my main interest. As a dentist, I started from there. When you look at this patient came to us at the clinic, and of course during my PhD, I wanted to do what we call a dental implant. If you look at the jaw here, actually there is no bone at all. You cannot put dental implant on this bone. So the only option is to have what you call to do bone transplant. But again, we don't have enough bone to be transplanted. Again, when you move the bone from the hip or the joint to the joint, then you create another problem on the donor side. And the failure rate is always very high also. Always there is rejection. And that is the starting. That's why we thought that we should try to introduce something new. And then we look at the literature. This is a very nice report done by one of my candidates. We've been in a 15th European workshop in periodontology in bone resorption or bone regeneration. And when we look at the literature, we could see that if you transplant the whole bone marrow containing cells, mesenchymal stem cells, in combination with biomaterial, we found that is more or less superior than the bone graft. That's again from the literature, which again is, we thought that this is something promising, that we are going in the right direction. However, to transplant the bone marrow, the whole bone marrow, if the surgeon or the clinician or the patient is not lucky or unlucky, you might transplant the bone marrow without sufficient number of cells. And that's why patients go to the hematologist for the bone transplant. They face the problem, sometimes they have to repeat the procedure several times because they take the transplant the bone marrow and then they found that they haven't got sufficient number of cells. And that is a problem. That's why we thought, okay, to solve the problem, we have to grow the cells in the lab, increase the number, collect the bone marrow, harvest the cells from the bone marrow, take it to the lab. Out of 10 ml or 30 ml of bone marrow, we could generate more than 100 million cells. And then we take it back to the defected site, if it is a bone or a liver or whatever. But then, how to do that? That's the question, because as I said, in the bone marrow, the stem cells are rare. In each 100 cells, 100,000 cells, there is only one stem cell. And that is a problem. So that's why we need to do that in the lab, to get the bone marrow, harvest the cells, and expand the culture. And our target actually to get at least 100 million cells within our bone marrow harvest. But even that, how to grow the cells? Yes, theoretically, it's relatively not difficult. But again, as I said, the cells are affected by the environment. So if you change the oxygen level, you change the behavior of the cells. And the outcome might be different. So you have to be careful. If you change add mechanical force, again, you change the behavior. So they are very sensitive. They can go in different directions. And that's why we need to always consider the condition where we are growing the cells. And that's what we are doing now. Good example. This is a postdoc from Japan in my team. He established this very nice model. We call it distraction osteogenesis in the mice. He used orthodontic appliance to pull the bone into two different directions. So in small, tiny mice, he created this kind of defect. And then, to repair this or reconstruct this defect, we use cells grown in normal oxygen tension, or we use cells grown in hypoxic tension, where the oxygen level is less. Just to show the difference between these two different conditions. And you could see after 3 weeks, the ones that the cells being grown in hypoxic condition behave better, and then we had better result using the same type of cells from the same source. One more thing that you have to consider the source of the cells. If you think bone marrow, I mean, if I'm talking about bone, I would prefer to use bone marrow cells. But if I'm talking about skin or fat, I would prefer to use stem cell from the fat. If I'm thinking about neurological disorders, the cells from the dental tissues are the best. Because of the source, because if you look at the embryology back, they are generated by different, they have different backgrounds. So do not throw away the, what you call it, the deciduous teeth. I think now we are talking about biobank keeping, try to keep the deciduous teeth, because it could be used to treat neurological disorders. Because if you look at the growth rate of the deciduous teeth even is higher than the cells derived from the bone marrow. But they are not the appropriate for bone regeneration. But for neurological disorders, it's perfect. And that is why, because their expression of cytokine and growth factors is different. So now we hear about stem cell, clinician, hospital, private, they are just taking mesenchymal stem cell, but without considering their origin of the resources. And that is not correct. You have to really think about the indication. If you are targeting bone, you have to use the right type of cells. And to show that, this is a comparison between cells derived from the bone marrow and cells derived from the adipose tissues. The expression of the cytokines and the markers is different. And the response also is different. If you use cells derived from the fat, the alkaline activity, which is a marker for bone regeneration, is different, is delayed. And again, demonstrated delayed osteogenic capacity, which means that they can make bone, but not with a good quality and might take longer time. So again, here is obviously, I clearly say that important to consider the source of the mesenchymal stem cell. All of them are mesenchymal stem cells, but they are not the same. That's important. I wouldn't say that I'm a pioneer in treating bone defect by mesenchymal stem cells. My collaborator in Finland in 2009, they treated this patient. You see the defect here is large defect on the palate. They use cells derived from the fat to treat this such kind of defect. And they consider the challenging issues related to the vascularization, because when you transplant the cells, if the cells do not have sufficient amount of blood, they will die in very short time. So what did they do? They collected the cells with the scaffold and implanted the cells in the stomach or in the rectum here, under the muscles, for 9 months to provide some kind of vascularization for the construct. And then after 9 months, you could see the outcome. Very nice healing on the palate. But that was a case report. It's not really clinical trial. It's only one patient. The same group in 2013, they treated this type of defect, ameloblastoma, after recession. And again, the outcome also was positive. They reconstruct the whole mandible by using cells derived from the adipose tissues, fat tissues, not even bone marrow. But again, adipose tissue is not the appropriate cells for bone regeneration. Another group in Netherlands in 2008, they reconstruct this defect by using mesenchymal stem cell from the bone marrow. But in their trial, they included 6 patients. Only the success was only related to 1 patient. So 5 patients being failed. And another problem that they use what we call it osteogenic supplement and FBS, which is derived from animal. And today, the ethical authority will not accept any animal product for human or for clinical application. So we have to develop something different compared to this protocol. So as I said, it's not possible to use animal product now. And then we thought that we should think about something else. We found that human platelet lysate might be appropriate and to supplement or provide the cells with sufficient nutrition. And of course, took me some years to work on this. And again, we managed to show that the platelet lysate represent very good alternative compared to the FPS, and even would help the cells to be directly towards bone-forming cells. So this has already been reported in 2020, and we could see that also we managed to optimize the concentration of what you call it, platelet lysate, which is derived from the patient. So it took me like from 2007 to 2019 to make a protocol for how to use stem cells in patients. And that is also published in 2019. And not with me, again, with a large group of scientists in Germany and in France that we work together to develop this protocol. But the protocol now is reported and it's been standardized and used commonly in Europe for all clinical trials. But of course, this is a challenging procedure because it's very expensive. You have to, I could see that now, which is really, not very happy to see that patients, they go to clinics to do stem cell treatment without considering these regulations. When you deal with the stem cells, I do a lot of research in my lab, but my lab is not GMP, not good clinical for good manufacturing practice. So to deal with human, you need certain requirement. You cannot expand the cells in a research lab, research grade lab. So you need this kind of laboratory where everything is quality control, contamination, all these issues should be considered. When I started the clinical trial, we did not have this kind of facilities in Norway. So I have to work with Germany. So I had to collect the bone marrow from the patient in collaboration with the hematologist, send the bone marrow to Germany, and then wait for 2 to 3 weeks using my protocol, and then get the cells back to Norway. And that is challenging procedure. There is a lot of logistics. When you send the cells through the custom at the airport, they should not be exposed to X-ray. It costs a lot. Someone has to take the cells with himself and go to that. So it's a bit challenging and expensive procedure. But this is the regulation. We cannot avoid that. Fortunately, based on our result, the authority in Norway, the hospital, they agreed to establish those facilities, and now we have the facilities in Norway. So when we get the cells back from the lab, we have to look at the viability of the cells, the stemness of the cells, characterize the cells, be sure that there is no contamination, and look at the efficacy also in small animals, that we assure that these cells are effective. And we get the cells in this kind of suspension. This is part of Reborn project. It's a European project. I was leading this project actually a couple of years ago, where I think we are 24 partners in Europe dealing with the same protocol. So we have done the validation, and we managed to follow the protocol that has been reported by us. But then we have to consider the delivery, how to deliver the cells to the body. If I'm considering talking about neurological disorder, for example, if I want to treat spinal injuries or something like that, I might need to inject the cells directly with the injection. But if I'm talking about bone, I might need a carrier, or what we call it, scaffold. A scaffold to provide the cells kind of room that they get the right information as they are in the body. Then the scaffold is another issue that should be also considered, that we should select the appropriate scaffold to deliver the cell to the defective site. And in our case, of course, this is a clinical trial. We are not able to use scaffold if it is not registered as a product in the market, if it's not validated. So we can't use scaffold just used for research. So I have to deal with a scaffold that is already being used by the surgeon or by the clinician. So we use this type of scaffold. We call it tricalcium phosphate scaffold, which is have the same property as the bone, containing hydroxyapatite and tricalcium phosphate. So it gives the right signaling, because you have to give the right signaling to the cells to instruct the cell to go to which direction. And of course, we had to be sure that the cells like the scaffold. If they don't like the scaffold, they will not attach to the scaffold. Then could be a reason for a failure. So we have looked at it, we have validated, and you could see that the cell nicely spread over the scaffold. So we had to look at this before we go to the patient. And of course, we had to determine the concentration of the cells, the number of the cells for the defect. If we are using 1 million cells or 100 million cells. Then we go through animal experiment. We use different concentration. We use this model under the skin of the mice, because that is what you call a subcutaneous model, where there is not a bone environment. So if we manage to make some kind of bone-forming, bone-like tissues, meaning that we create some kind of construct, we call it osteoinductive construct, that could make the bone. So we could see that we reach certain concentration, we compare different concentration of cells, and then we could make nice new bone by using 20 million cells per centimeter square or 1 millimeter. So if you consider 5 ml, then more or less we'll go to the 100 million cells per 5 ml. And that's the concentration we agreed to use it for our clinical trial. But then again, I got a question from the ethical authority in Norway. They said, Kamal, this is considered as a drug. It's not a biomedical device. So they deal with it as a drug, as a new drug. So you have to think about the safety issue before the efficacy. And then we need data from large animal. We cannot take it from a small animal, because it's something new. It's not even published in the literature. Then my surgeon had to travel to Italy, work with another university. We use mini pigs, because the pigs, more or less, their healing time is more or less comparable to the human. So we have done the experiment in mini pig in Italy, just to collect some data to convince the authority that can be safe and effective. So we thought that let us try to reconstruct the jaw bone, the alveolar bone. And then we thought, okay, let's make tiny alveolar bone. Cut the bone, make it very thin, and try to reconstruct it. If it works, then we can do the same on the patient. So we cut the bone, generated very small, thin bone, alveolar crest, and then we used the scaffold or the bone substitute material that used commonly by the surgeon to make new bone. We thought that if this is going to work with only biomaterials, then we might not need the cells. So we placed the biomaterial commonly used by the orthopedic surgeon and the oral maxillofacial surgeon on one side of the animal, one side of the jaw, and then we used the combination of the cells on the biomaterial on the other side. And obviously you could see with only biomaterial, we had failure. We did not have any new bone. On the other side, nicely newly bone being formed. And even we managed to put 2 dental implants into this newly developed bone. So this has been successful. We managed to convince the authority that this is something can be taken to the clinical setting. But then, questioning back the question of Saeed about the regulation. To get the approval, I had to process what we call it voluntary harmonization procedure. I had to get the approval in 6 European countries at the same time. And it took me 3 years to do that. Because the regulation in Germany is different from France, different from Spain. But it works, because and even the Norwegian authorities were happy to take the lead, because it was some kind of new exercise for them. But we manage. And we manage, and then we could continue. But just something important. So the indication for the first patient that we should select it, a patient where the patient came to us with very tiny, thin alveolar bone. The width is only 1.5 millimeter. And if you think about the diameter of the dental implant, it's between 6 to 7. So it's impossible to put dental implant in this tiny alveolar bone. So you have to reconstruct the bone. And we thought this is challenging, even challenging for us, but we thought if this is works here, then it can work on other type of bone defect, even on the long bone. Considering that this is the most contaminated area. If you think about the oral cavity, there are thousands of bacteria on the oral cavity. The blood supply is not sufficient. So if it's going to work here, then it can work on the other bone defect. And more than this, from ethical point of view, we would be able, after the treatment, to collect bone biopsies, because at the end, we want to put dental implant. So while doing the dental implant, we have to make some kind of drill on the new bone, and then we collect biopsy. But if I treat only fracture on the long bone, if the treatment is successful, the ethical body will not allow me to take biopsy from a healthy new bone. But then again, that's model that we should, at least for proof of concept, that should be the right model. So that was the first patient in our trial. And again, as I said, the recruitment criteria, the patient that recruited in the trial should not have more than 4 millimeter or less bone in the alveolar bone, and they should be more or less within the age between 18 to 80 years. If you look at the CT scan, you see the resorption, severely resorbed bone. As I indicated or mentioned, we collect from the cells from the bone marrow, from the hip, send it to the lab, get it back in the surgical room in these 2 syringes as suspension, 100 million cells, 2 syringes of the cells, and then the scaffold on the other syringe. We connect the 2 syringes together to avoid the contamination. And we do that as a surgical room, as operation room. I don't know if some of you are if you have interest, that this is the surgical procedure is very normal surgical procedure. It's not difficult. The challenges is more or less related to the production of the cells rather than the surgical procedure. But this video shows that we prepare the defect site. We put this kind of membrane to keep the scaffold and the cells in the right place. So this is the mixture of the cells, and together with the scaffold, which is in the form of a powder, and then we cover everything with a membrane. So we follow the contour of the alveolar ridge, so we don't get overgrowth. That's something important also. And then we do that and let the patient go and come back after 5 months, which is the normal healing period for the bone. When you think about bone formation, you have to wait, for a couple of months before you get the new bone. So before treatment, 1 week after treatment, and 6 months after treatment. With CPCT, again, you could see we managed not only to reconstruct the bone on horizontal direction, but also in vertical. With bone graft, it's impossible to reconstruct or to augment the bone in a vertical discussion. I think the surgeon agree with me with that, which is also very nice new finding. And you could see from this cast, You see the width before and after. I think you agree with me, this is very clear result and promising. This is before and after. We managed even to install two dental implant. We collected the biopsy, histology, you could see formation of bone around the scaffold. So that was promising. Before treatment, you see the dental implant installed in the newly developed bone. But more interestingly, you see the nice soft tissue healing. This patient is 70 years old patient. And when you look at the gingiva here, if there is a dentist here, he would be more or less impressed by having this kind of. Although the cells were not in direct contact with the soft tissue, because it's being completely covered. But then which more or less indicate the paracarine effect of the cells. The cells also, they send their signals far away from the site and influence the healing of the soft tissue, which is really amazing. And you see the patient got new teeth and new dental implant. This is after 5 years of follow-up. Another interesting finding, with bone graft, usually we get bone resorption. With the stem cell treatment, you see there is no resorption after around the screw. Again, that is another interesting finding, which would require further investigation. Then in this trial, we treated 11 patients successfully. All these are biopsies, the newly developed bone. So I think we thought that maybe we are lucky. I think that was a very good result. We reported to the EU, and the EU also accepted the result. All the patients were satisfied. They got their dental implant, their crown. We had some kind of questionnaire. Most all of them were. They said yes, they're satisfied with the treatment. And when it is reported from this 11 patients, we managed to convince the European Commission to expand it to moving from phase 1, phase 2 clinical trial to phase 3 clinical trial. Because in phase 1, usually you talk about safety only. Even if we have very good result, effective efficacy, still we cannot talk about efficacy because there are limited number of patients. But we have to go through this step by step. From phase 1, phase 2, then we move to phase 3. Phase 3, which is more or less meaning that we should have a control. In the first clinical trial, we didn't have a control. Here we have the control, which is a bone graft. And then again, it's also a multi-center clinical trial. So we had to include other surgeons, not relying on the surgeon in Norway. And that's why we got another grant from the EU, and we recruited 150 patients. 100 with stem cells, 50 with bone graft. And of course, we had to calibrate. We have to train all the surgeons to more or less to follow the same procedure. And again, 6 countries, European countries, were being involved. And after 60 patients, we could say there was no major adverse event, because we have to report to the authority, make sure that there is no adverse event. This is my colleague from Madrid, Professor Mariano Sanz, and this is his patient. Again, he managed to get the same result as the result generated in Bergen. Histology, again, being done in France with the same result that done by our pathologist or histopathologist. So we are happy with that. In this trial, the first trial, we treated only the lower jaw. In the second trial, we included also maxilla. And again, also, you could see nicely developed bone where we had also dental implant in the newly developed bone. And we could say the outcome clearly says that if it's not superior than the bone graft, it's equally good as a bone graft. So this is, I think, is very good result, promising result. Recently, it's already been published also. You could find it, and it's published in, I think, in August. It's a big clinical trial. So it is promising. However, the cost is relatively high. For each single production, it might cost around 20,000 euro. This is expensive because we had to grow the cells in GMP facilities. But we are working hard to reduce the cost. One way to promote or induce promote the growth of the cells, giving them better signaling, using mechanical force, for example, using this bioreactor system. This bioreactor system also developed by one of my postdocs from Japan. That might reduce the cost. Based on this, we also managed to treat long bone defect in collaboration with the orthopedic surgeons. So we treated non-healed union after complicated fractures, and actually we treated 20 patients together with the orthopedic surgeon. Good example, this patient is a boy of 12 years old. He came to the hospital with a multiple fracture, and they repaired or did the surgery. It turned out that the left leg was shorter than the right leg. So what to do? He can't walk like that. Then they tried to do what we call a distraction osteogenesis, pull the two sides of the bone and fill it with bone graft in a way to increase the length of the left leg. And they did the surgery 3 times and unfortunately failed 3 times. So the patient could not walk for 5 years. And then the orthopedic surgeon from the hospital, they called me and said, Kamal, you are a dentist. You managed to treat bone defect in the oral. Let us try your protocol before we. And then we said, okay, let's try the protocol. We provide them with the protocol. My surgeon, the oral surgeon, worked together with the orthopedic surgeon. So you could see that after 6 months, we could close the defect. And they remove all the plate. And the child, this is the child, you see? At the age of 17, he started to walk normally. So it is promising. Of course, still you have to find the right appropriate scaffold, because the scaffold available now for us is this kind of granules, which is a bit challenging to work with this scaffold. So we need to use another approach. So in my team also, we are working with 3D printing. We'll try to mimic the bone by making scaffold more or less look like the bone using the 3D printing technology. So this is already ongoing research. It's not yet at the level of the clinics, but we got 4 patents actually out of this idea. Another issue, that is another approach, that is do what we call bioprinting. Because in our protocol, we implant seed the cell normally, just normal cell culture condition. We just use a pipette, whatever. But then we have challenging that we will not be able to control the distribution of the cells, the number of the cells, and all these things. But if you manage to print the cell directly within the hydrogel and consider and more or less control the number of the cells, distribution of the cells, and you could even use more than one type of cells. You could mix mesenchymal stem cells with these endothelial cells and have them in 2 different layers, for example, 3 different layers. So this is also something that is, this field of bioprinting is really growing rapidly now. So you could bioprint everything. All organs can be, you could really, and now with the AI, you could design the whole construct and just use the robot and make your construct by using the bioprinting. This approach is still working, is growing, but we manage so far you could see that this is the stem cells on the scaffold being bioprinted, and they managed to survive for 3 weeks without any problem within the bioprinter. So it's something that I think soon that we will be able to develop protocol based on bioprinting. But there are, of course, some challenging. We need to address it. Like, for example, we have to mimic the metabolic needs. For example, how to design the right architecture. Then we use the AI to design the construct, which will save time, this error and trial and error. So by the AI, we'll be able to do that in short time. Still haven't done that for the patient, but I have submitted the grant application to the EU. If it's accepted, then we'll go in that direction also. Everything costs money. Without money, you can't do it. So to summarize, this is just a short video to summarize what we are doing. No sound. So we collect the bone marrow from the hip. We take it to the lab. We grow the cells on the scaffold. If you think this is the right stem cells. The cells like to talk to each other within the scaffold. They produce their cytokines and growth factors and make their own network. And then we bring everything back to the defected site, implant it in the defected site, and then within 4 to 5 months, we get completely new bone. So to conclude, I can say mesenchymal stem cells is, as a treatment is a promising approach and is developing very rapidly with the technology now, with the AI, with the automation, robotic technology, and all these things. I think in very short time, already now we are in phase 3 clinical trial, meaning that we are in short time we could go to phase 4, and then we'll have the companies on our side. Then we'll be able to introduce it as a routine treatment in the hospital. You talk about multidisciplinary, you see, these people are working with me from all Europes. More than 40 institutes, actually, who are working together, institute and companies. It's a relatively large team. Funding, of course, I should always acknowledge them. Not only 5, I think there is one missing. The whole project is covered by 6 European projects. So we are happy that we got funding 6 times from the EU Commission, but also from the Research Council of Norway, from funding agents, local fundings, supported by the companies also. I think by now, I think it's on time. Good. I'm happy to answer questions. Starting from your last question, follow-up, yes, as you see in one of the slides, I presented 5 years follow-up, but now we have 8 years follow-up from the first patient. And it's not been reported, but I think soon we'll get 2 or 3 papers coming out of follow-up. So I think the most interesting finding that there is no bone resorption, which is absolutely interesting finding. About the cancer, yes, there is debate, but not related to the adult stem cells. It's more or less related to the embryonic stem cell, because there is embryonic stem cells, yes, that can be considered. It might be carcinogenic. This is not proved, but there is a question. But for adult stem cells, there is shouldn't be worried about that. Now we started with more or less physiological defect, but our ultimate goal, of course, to target bone defect related to cancers and patients that have been treated by chemotherapy and like that. But again, we have to take it step by steps. We cannot solve all the issues. But our ultimate goal is the large defect after recessions because of the cancer. Umbilical cord, yes. As you see, my group is relatively large. I have some of them we are working with umbilical cord. Some of them are working with dental stem cells. Umbilical cord is very good source. But from my experience, from our animal experiment, is not the appropriate cells for bone forming cells. Hematologies are the best, because our wasted material is easily that, and they are stem cell. So they express all the marker for the stem cell. But for bone, we look at them, we compare them, still they are lower, less effective compared to the bone marrow cells. But again, depending on your indication, you have to choose the right source. Umbilical cord, yes, absolutely, yes, for several indications. No, not like that straightforward. That's why I had to do what we call this voluntary harmonization process. Then everything is harmonized. But if you do that, you don't have to do that. You can get the approval separately from each country, which is rather less problematic. But to do this harmonization, then it's already approved in all of them. But it might take longer time. But two different ways.
Thank you very much, Professor Kamal, for this outstanding lecture. And you are really one of the eminent scientists in this field. I have 2 questions concerning this. You showed in one of your experiments that when you grow cells in hypoxic condition, they showed better proliferation of the bone. So is this like, is there any mechanism behind this? Genetic modification happened inside the cells during the replication of the cells in the median culture? Or this is like, there is other reason behind this? The second question, general question for this approach, is there any risk of graft rejection during the implant of?
Risk of?
Graft rejection. Since it's like as a stem cell, so just like this in general.
Yeah.
Prof, thank you.
Yeah. The last question, rejection is, I mean, you could see from my patient that we don't have any rejection, no even adverse effect. But if you think about the mechanism, how does it work? As I said, the stem cell can be differentiated towards the right direction, can be made more or less considered as bone-forming cells, or they can secrete cytokines that influence the endogenous cells. And what they do, they can modulate the immune system. Because mesenchymal stem cells, they consider as immunomodularity cells. So it's not only their differentiation capacity. So they play or they manipulate the immune system by itself, which means that they will control the inflammation. And that is, for this principle, we use this principle to treat our multiple sclerosis patients, because the multiple sclerosis is inflammatory disease, isn't it? And then we don't consider that the cell will be derived or differentiated to be more or less like brain cells, rather than just control the immune system. So this is the way that so rejection, yes. Again, they express very low MHC, which is, and express also, or what do you call it, protein, suppressive protein. Hypoxia, again, because of the, there are certain, this is a complicated mechanism, but you have to consider hundreds of thousands of cytokines and protein and like that. And oxygen tension, of course, influence all this production of the cytokines, and that might, because as I said, when I started the presentation, I said the microenvironment, the environmental condition, including the oxygen, would influence the secretion of the cytokines and chemokines. And that would influence the whole mechanism. So for bone, yes, it works better. Welcome.
Thank you very much for your very nice talk. And I have one question about the use of colony-stimulating factors during the in vitro growing of stem cells. Do you
Growth factor.
Stimulating factors? Do you have been used?
No.
Because in the dendritic cells or other cells which we are using in other tissue cultures, we have to use colony-stimulating factors or something like this.
No. We did not use any growth factors. We tried to not manipulate the cells.
Only.
So that is the difference.
Hmm.
If we use adipose stem cells for bone formation, you need to add BMP2, which is growth factors more or less induce the bone formation. But for the bone marrow cells, you don't need to do that.
Yes.
So when I talk about this supplement, which is in our case, is 5% of platelet lysate. It's not a growth factor.
Yes.
And that is used only during the transport of the cell from the laboratory to the clinic within 24 hours.
Hmm.
So it's not for the growth of the cells.
Yes. Because I attended one trial in North Carolina, and we working on developing or growing stem cells for kidney,
Yeah.
and they use progenitor cells.
Exactly.
Yeah.
I know different groups. I mean, you could use fibroblast growth factor, for example.
Yeah.
But again, there's always question about the growth factors, and there are some. It's not always possible to control the outcome, and I think there are more ethical consideration. So we try it again for at least for the patient. I mean, for the animal, we use growth factor in our experiment. That's not a problem. But when we wanted to do it in the patient, if we manage to increase the number of the cells to 100 million cells without using growth factor, why should I use growth factors?
Yes. I got it.
Yes,
Thank you
simple
very
as
much.
that.
Thank you.
Thank you very much for your informative and interesting presentation. Great work indeed. I have a question. When you try to determine the dosage per patient, how much does each patient require in terms of how much stem cells and scaffold? Does it go by area, centimeter? And have you had cases where you had to do maybe to osteonecrosis or anything of cells that you had to overdose, grow more cells into the scaffold? That was like
You are quicker than me. This is my ongoing project now is targeting osteonecrosis patient.
Yes.
Because as I said from the beginning, I thought that the best that we target physiologically healthy, patients that without disease for the proof of concept. But now we have done phase 3 clinical trial.
Yeah.
Then we thought it would be better that now it's time to go to the disease patient. One of them is osteonecrosis. And osteonecrosis is a challenging issue. It's the infection is one thing, but the regeneration is another thing. So you have to control the infection on one side, and again you have to regenerate the tissues of the other side.
Yeah.
But this is ongoing projects.
Okay.
In animal, I can say that we have very good result.
Oh, great.
We haven't started the clinical trial now, hopefully by next year.
Okay.
We are preparing now. We just submitted the application to the ethicals. As soon as we get the approval from the authority, then we'll start with treating the.
Oh, thank you.
And this is not mentioned in my last slide. This is one of the also new EU projects.
Oh, okay.
Because I collaborate with Liverpool University. They are also developing new scaffold
Okay.
based on pectin derived from potato. You see? Because potato also shown that it has some kind of molecule that would more or less target or resolve the inflammation. And we thought that would be the right scaffold for this kind of group of patients.
Okay.
So we have completely now new European project focusing only on osteoporosis, and we are 15 partners in Europe.
All the best. I just have a question out of curiosity. How long does it take till you isolate the cells, till you characterize them and all that, before you do the transplants? Before, yeah. How long does it normally
The
take?
protocol for the not characterization, you can do it in 1 hour. That's not a problem.
Okay, with the flow.
But
yeah.
the expansion,
Yes, how long?
to
does
reach
it
this
take?
100 million,
Mhm.
we tried 2 different protocols. The first trial, we used 3 weeks protocol.
Okay.
And that is also related to the logistic, because we send the bone marrow to Germany, we take it back. We try to avoid the weekends because the scientists, they don't work during the weekend. So this is a lot of challenges, issues. I didn't have to talk about that. But the cost is rather high. When we go to the second trial, we saw that maybe a good idea to reduce the cost by reducing the expansion time from 3 weeks to 2 weeks.
Okay.
We managed to do that, but we had some challenges. I mean, we did not get a sufficient number of cells in some of the patients. Because
Okay.
again, you have to think about the individual variation. So not all of them are standard. You get 100 million cells from
Yes.
all the patients.
Mhm.
And that's why the 3 weeks protocol is more safe compared to the 2 weeks
weeks.
protocol.
Okay.
However, of course, in the future, we're trying different things, the bioreactor and all these kinds of things, that more or less we might be able to come back again to the 2 weeks protocol.
Okay.
But at least you need 2 weeks to have sufficient number of cells.
Okay, thank you very much.
Happy to answer all these questions. That means it's quite.
Assalamu alaikum.
Alaikum salam wa
Thank you
rahmatullah.
very much for the excellent and impressive, I wouldn't say a project, I would say a humanitarian and novel global rescue mission. All the best to you and your team, and I wish you all the best expanding this project.
Thank you.
There are so many questions. I tried to put them in one after the other, but I want to give the chance to others as well. I have 2 questions, major ones. The first one, have you looked at the predictors for successful, for a success procedure? You mentioned the environment and how the environment could influence the donated stem cell, but have you looked into the indigenousness characteristics of the donor that could affect the success? And there's one reason why I'm asking this. While you were presenting, I was thinking about the situation in Oman, and not that the oral health is good, but I'm a physician. I have other concerns in my brain that are priority to me, like diabetes, diabetic foot, ulcers, and so on. So what would be the predictors or factors that could be considered as a success factors to influence successful trials or operations or transplants? The other question, you mentioned the handling procedures and the logistics that involves traveling and bringing and putting and all of that. I'm sure it's there in your protocol, but then how do you ensure the quality control measures are put in place? And also, in case of violation or suspecting violations, how do you go about investigations and penalties? Thank you.
Very good. of course, prediction is very, very, thank you for this question. I mean, maybe if you go back to my first clinical trial, we recruited 13 patients. We managed to treat 11 patients. So 2 patients that has to be excluded from the trial, not because of the treatment, because that we did not have sufficient number of cells. And then that would lead to your question, if this is the right individual or not. Exactly. And if there is any way to know that before you harvest the bone marrow, because it's, again, is invasive procedure to get the bone marrow from the patient and put the patient in the trial and then say after 2 weeks, sorry, it doesn't work. So if you have a marker, biomarkers to tell you, but this is one of my new projects. We try to get this information from the peripheral blood, from only just getting blood, try to define or identify certain biomarkers. Together with the AI will be able to. But this is just, yes, this is a very good question, and we are working on that. And I think that is important because always there is individual variation. I have another project that more or less I'm moving from autologous cells to allogenic cells, establishing a biobank. If we manage to do that within the coming 5 years, then you could send me and say, Kamal from Oman, I need cells from your biobank. I can send it directly to you. And I say, this is effective and safe. So this is also ongoing project. So yeah, again, developing is rapidly, but I'm quite sure in some few years we'll be able to answer all these questions. But to me, future is allogenic cells. I did not address in the lecture. Also, you could use only the collected proteins from the stem cells, what we call it condition medium cocktail of condition growth factors driven by the cells, exosomes, for example. So there are a lot of things ongoing. So it's not only the transplantation of the cells directly. So the second question was about quality control. Of course, this is we have very strict protocol. It's not by us, by the authority. So I am the sponsor of all this clinical trial. If the surgeon in Germany gets the cell from the laboratory, he or she would not be able to transplant the cells without my approval. So I have to be like online on 24 hours as a sponsor to say yes or no, because there is certain criteria for quality control. We call it release certificate. So we have certain release certificate. Number of percentage of, for example, CD90 has to reach 90% stick. If it is not 90%, it's out. Contamination, there is all this is going through all this. We have to go through all this per patient. Then I would allow the clinician to use the cells. And it happens sometimes if I'm not satisfied with the quality control or the stemness, not only the infection. I mean, even the stem cell. If I'm not convinced that is a stem cell by more than 90%, I would not allow the clinician to do that. And it's a responsibility, and I can be taken to the court because of the authority. So there is very strict regulation about this.
Any other question? Yes, Dr. Hussein.
Thank you, Doctor, for the informative presentation. Yeah. My name is Dr. Hussein Al-Amri.
Yeah, hello.
My question, is there any, before we start transplant, is there any lab test other than HLA test, which is very important here? And is there any side effect or maybe the risk might the patient have?
On the bone trial, we didn't have any adverse effect. So we treated more than 100 patients, and there is no adverse effect because I said we have to report to the authority. in Europe and say that if there is any reaction from the harvest of the bone marrow or from the surgical procedure or even after the treatment. Within the trial of the multiple sclerosis in collaboration with the neurological disorders, I think there was some kind of reaction because the cells being injected directly into the spinal cord. And there was, in 2 patients, created some kind of inflammation observed by the neurologist. So they have to deal with that. But there was no really serious adverse effect because of the treatment so far. We do, I mean, of course, we have, for example, the microbiology lab has to be part of it, for example, that to look at all these microbes and all this kind of, we have the, what we call it also the, HLA, of course, that is, so again, there is long list of certain criteria, you have to do it. And so again, as I said, this is a multiple, so we have to have all the laboratories on our site, and they have to collaborate with them. So some of them on site, some of them we have to send the samples to look at the quality of that. And one of the most important issues is the markers for stem cell, if they are stem cell or not. So there is certain, we call it release certificate. So there is a long list we have to tick on all in each items by the lab. Immunophenotyping also, we have to do cryotyping for it. All this has to be covered.
Yeah. Nabras al-Harmi here at the back. Assalamu alaikum.
Alaikum assalam wa rahmatullah.
Just to bring up the same question, because Dr. Hussein bring it up about the HLA, and Dr. Thamra also mentioned about successful factors. I am wondering if you are interested on explore the genomic side of individuals as a prediction of the output. And if you have explored that area, what could be the expected output?
Of course, this is one approach. Absolutely, the approach, we have to look at it, of course.
Have you ever looked at that side, and what is kind of output you get?
We haven't looked at it as a prediction factor. That's we haven't looked at it. But of course, this is something that we should look at it, of course, in the future. Because our mainly focus now more or less on to proof of concept to show that this is safe and effective. But then the next question is the donor and if this individual is the right donor or not. And of course, when you want to establish this biobank for allogenic cells. Now for autologous cells is less important because you are dealing with the same patient. Dealing with the same patient, collect the bone marrow from the same patient, treat the patient with the same cells. But if you want to use allogenic cells, then you have to consider all these kind of factors. If you are using the right donor or not, because we don't want to use wrong donor. But it's a good question, of course, but something that has to be considered.
Yes, first of all, thank you for the wonderful presentation and for sharing your experience with us. And I'm so happy that maybe this is the first time I heard that we are highlighting this topic and talking about stem cells, especially here in Oman. As I recently graduated with a master's degree in regenerative medicine and specialized in stem cells. And when I'm trying to attract the other people and research, for the research, I didn't find anybody who's cared for this, or highlight this topic about stem cells honestly. Regarding your presentations, I did observe that you obtained the mesenchymal stem cells from the bone marrow, isn't it?
From bone marrow.
Bone marrow, yeah. Which is it autologous or allogenic?
For the time being, it's autologous.
Yes. Okay. So we found that, or we know that the mesenchymal stem cells exist or reside in many, we can say, tissue in the body. If we are focusing about the teeth, it is reside or exist in the dental bulb.
Yeah.
Okay. And we did So we are talking about the adipose tissue also when we compare that the osteogenic differentiation or the capacity of the mesenchymal stem cells to the bone or to the bone. Yeah, I was working in a research before comparing the differentiation capacity of the between the bone marrow and the adipose tissue. Why we didn't refer to the adipose tissue since it is less invasive procedure than the bone marrow?
Yeah. Yes, the adipose stem cells is definitely less invasive procedure. There is no doubt about that. You could see that in my slide, the first pioneer group working with adipose stem cells is the Finnish group from Finland, from Helsinki. They have done several cases, but now we convince them to go back to the bone marrow. Because, of course, it is mesenchymal stem cells, but as I said, they are not inherited for bone. I mean, the origin is not from the bone environment. And the expression of the certain cytokine influencing the osteogenesis is different. So you might have delayed healing. You might need growth factor like BMP2, which is not needed. So adipose stem cells, less invasive, but I wouldn't recommend them for bone. I would still prefer to use bone marrow, and then people that they've been working with adipose tissues, now they are convinced that they should not use this for bone formation. Dental stem cells are good resources, but again, mostly it's appropriate indication for them is neurological disorders, not for the bone.
Thank you so much. I apologize because of the time. I know so many questions, inshallah. I would like to take this also opportunity for me to ask question. If Dr. Fahad, just, inshallah, please. Now, do you think age-wise play a role in the process of success of this regeneration? Number two, what is your prediction about using this in enamel, dentine, to recover from dental caries? Because I know King's College, they're doing such studies, but until now, I know they're not successful. The last question, you are with us here. We, and I'm sure maybe Dr. Fahad would agree with us, it's very interesting if we could do some collaboration, Oman College of Health Science with you guys. And I'm sure the lab and pharmacy, maybe they'll be interested, يعني. سامحوني, I took the advantage.
I mean, for collaboration, I'm very positive, very happy to collaborate with anyone from Oman in general. Of course, all of you are my friends. Fahad, we've been friends. I mean, this is developing institute. I would be very happy to collaborate. That's not a problem. You look at our website. Also, there is a lot of things that you could look at it where you could find common areas that we could collaborate. As I said, my umbrella as a leader of the Center for Translation Research, I have teams that work with public health, team work with biomaterial, team work with inflammation, teams working with different types. So we could find the right person to collaborate with. So collaboration, don't be worried. As long as that we are able to generate joint project, I would be happy to support it. There is no doubt about that. Be sure about that. Using stem cell for enamel is a bit different, of course, because it's not like dentin or pulp, of course. I have two of my researchers are working on dental regeneration. And if you look at the Japanese group, they are ahead of us using that. They managed to develop complete tooth now. I think that's already been published. The only difference that the occlusion was not really good occlusion. The tooth come out from in the mouth in the rat was smaller compared to the natural tooth. So there is heavy ongoing research, not by me, but by other groups. So that is also possible.
What about age-wise? Do you think
age? You see my patient, I didn't say anything about that, but they are coming for dental implant. And in Europe, someone come for replace it or ask for dental, you would expect that this is a senior person. So most of my patients were above 50 years old. But it is true. when you think about the growth of the cells, is different from young patient to senior or old patient. But I don't know if by chance, most of my patients are, some of them are 70, 75, and 80 years, and it works. So if it works for all patients, it would work for young patients.
Thank you so much, Prof. Kamal, صراحة, for the insightful and wonderful presentation. I really appreciate you, and I'm sure everyone have enjoyed listening. صراحة, ما شاء الله, يعني. Please applaud him.
Thank you.
I would like also to invite Dr. Fahad and Dr. Hasna, please, to jointly award.
I should come down to you, Fahad.
لا 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 ما يصير لا ما يصير هم بيجون
I didn't like this.
عشان تصوير عشان تصوير.
شكرا.
Very good talk, Salah. Very nice talk. Yeah. Just maybe I can give.
تفضل دكتور.
Thank you very much, Prof. Thank you very much, Prof. Kamal. As you know, we know Prof. Kamal for a long time ago, and I always tell him that you're Omani. He's a Jabir bin Zayd graduate. Even his brother, Dr. Hussam, also is a Jabir bin Zayd graduate.
Yes.
Thank you very much. And I know Prof. Kamal is very generous. I mean, he's been collaborating with Oman. I mean, maybe first time with the Oman College of Health Science, but already scientists from Oman already visited his lab. I mean, for research in stem cell. And for people who are working with stem cell, yes, there are people working with stem cell in Oman. We have an SQU, we have a bioreactor for expansion of cells. Nizwa University also have a center for stem cell research. But I think this collaboration, inshallah, will work on, inshallah. So we have some appreciation for you, Prof. Kamal, inshallah. Thank you very much, Prof. We have more collaboration, inshallah.
Thank you. Thank you.
Pleasure. Thank you so much for your attendance, صراحة. You made even the presentation, the talk wonderful. Thank you so much. Well, see you next time, inshallah. Bye bye.
Yeah. I hope. Okay, I'm glad that it was.
Yeah, more in medical imaging.
Medical imaging. I will try to link it with the modalities.
Yeah.
Imaging
Yeah.
modalities, maybe.
Yeah. Good. Alhamdulillah.
شكرا جزيلا بروف صراحة.
لا شكرا.